Well, welcome everybody to the Tree Fund webinar series, our second webinar of 2026. It's wonderful to have everybody back on here again. As y'all know, my name is Bo Broadbeck, and it's also always a pleasure for us here at Auburn University Extension to host and moderate this webinar series. So thank you for joining us once again. I think we have a great speaker, a really fascinating entomology topic for you all today. And as is our tradition, I would like to introduce my co-host, Heath Hupke with Tree Fund, to give us a little bit of a Tree Fund update. Welcome, Heath.
Well, thank you. Thank you, Bo, and thank you all for attending today and welcome as you keep popping onto our webinar. This is our second webinar of the year. If you missed last month, please go to Tree Fund's webinar archive page, and you can get a link to last month's wonderful webinar that we held at a different time than our typical one, and we will continue to experiment with that throughout this year. But I do want to also say thank you to our sponsors. That we have many sponsors, organizations, and people that support the work of Tree Fund, from our operations to building endowments so that we can fund wonderful research like we have today. So thank you to both our Crown, Platinum, and all those that have been able to donate throughout the years. And for many of you, if you're not familiar, at Tree Fund, we are an endowment trust. So we're not supported by government funding. It is purely on the donations of people and organizations like yourself. One of those opportunities that we're able to have, and I know this is probably small for a lot of you, and it's a great thing that it's small because we have many scholarships that we are able to support the future of urban forestry and arboriculture through undergraduate scholarships of $5,000. And we still have our current scholarship application open. So if you are a student, or you know of a student, or even a college, university, or program that may have students that would like to apply, we have up to 10 $5,000 scholarships available. The application is due on March 15th, so just under 2 weeks, but we would love to get more of our future involved in Tree Fund, involved in their programs, and support them financially as we are able to. So we're very proud of our various trusts, endowments, and organizations that have come and said, we want to support the future of this field, and we want to make sure that there are healthy $5,000 scholarships available for these young minds. So thank you. If this is something that you're passionate about and you would like to donate, we're always accepting donations. We're happy and thankful for all those that are able to support Tree Fund in this way. Your tax deductible donation is tax deductible, so we would appreciate that. And if you have any other questions, feel free to reach out to myself. Kind of logistics for today. This is a 1 hour webinar. We ask that you attend the whole thing. Stay online, listening to the question and answers at the end. And if you do have any questions you have, there is a button, should be probably at the bottom of your screen, that you can put in there and then we will get to those at the end. When you register today, we already have your ISA information. Tree Fund will submit that. And right now we have over 800 people at this webinar. So it does take a while for ISA to get through all of those CUs. So please show patience. But if you have any questions along the process, reach out to education@treefund.org, and we'll get back to you as soon as we're able to. And lastly, right now we do have 2 webinars upcoming that are secured. We are working on a June date, but right now in April 21st with Dr. Richard Hauer on tree health implications of long-term strategies to treat emerald ash borer. And then on July 21st with Dr. Thomas Whitney, the efficacy of aphthalates. Ooh, I'm not even pretending like I can say that. I'm not qualified to say all those words, but please look on our Tree Fund webinar page and feel free to register for those. And next, I'm very happy to introduce Dr. Caleb Wilson. He's an assistant professor at the Urban Landscape Entomology at the University of Kentucky. He's originally from Illinois, and as a researcher, he studied pollinators in urban farms, scale insects on urban trees, and emerald ash borer and post-invasion forests. Caleb's lab is at the University of Kentucky, where he studies sustainable insect pest management in urban landscapes. Current projects are focused on developing action thresholds and management strategies for scale insects on urban trees. And we also have Zoe York, who I believe is working on this project now and in the future. So, Caleb, I'm going to pass it on to you.
Alrighty, thanks, Heath. So I'm just gonna, is my screen sharing right now, or do I have to do that on
Not
my end?
yet. You'll need to reshare it.
Okay, gotcha. Alrighty, look good.
Yep. Go to presentation mode.
All right, perfect. Okay, just yell out if something goes wrong because I can't see you guys on the side here. All right,
Sure.
folks. Well, thanks for coming to this webinar, and I'm glad to see that we have so many people from all over the world here to learn a little more about scale insects. So I'm really looking forward to chatting with you all about this project. And so, as the title suggests, we are looking at a novel way of controlling scale insects in urban trees by disrupting these mutualisms that they have with ants. And I'm just going to take a minute here to hide this bar at the top of the screen. Um, Hold on a second, folks. All right. Well, it looks like that's the best I can do. Okay. So, oh, there we go. Okay, so as Heath said, I'm an urban landscape entomologist. I'm based at the University of Kentucky. And this is just on the left here, I've got a list of some projects that we're doing right now in the lab. A lot of our work is focused on scale insects. And so that's what I'm talking about today is one of many projects that we have ongoing with scales. We've also done a lot of work well, not so much here at Kentucky, a little bit work here at Kentucky, but before I came here, I did a lot of work up in Michigan with emerald ash borer. We still have one project that's kind of wrapping up here at Kentucky. And then a new focus for the lab is we're branching out into doing some turfgrass research. And so now we're doing some work with the annual bluegrass weevil on golf courses here in Kentucky. And so if you want to learn more about the projects that we're doing in my lab, this is my lab website. I'll show it again at the end of the presentation, so don't feel like you have to write it down right now. It's just my name with dashes between my middle initial. And so before we get into talking about this project, I want to just thank my lab technician, Zoe York. She's also a PhD student in my lab, and she basically runs the day-to-day operations of the lab. She is the lead person who did this project that I'm going to be presenting on. It's a part of her PhD dissertation. I'm just kind of talking overall about the work that she's been doing, but she was the person doing the boots on the ground work for lots of the things I'll be talking about. And then, of course, this is a photo from our lab group from last summer. We are a small but mighty group of researchers, and this includes undergrad students, graduate students, and some technicians. So a lot of them helped out with this project as well. Okay, so this is a presentation about scale insects. There's tons and tons of different species of scale insects, and depending on where you live in the world, you may be intimately familiar with certain species. Lots of scale insects have a pretty broad distribution, but they tend to be really pestiferous in certain locations in the US, for example. Typically, it's thought that, where their climate is ideal is where they tend to explode in numbers and become really common or where their climate is ideal for their growth and development. So where I'm at in Kentucky, these are some of the species we see fairly often. Things like magnolia scale on your magnolias, oak lecanium scale loves oaks of various different species. Crepe myrtle bark scale is not too common here, but down in places like Alabama or Texas, crepe myrtle bark scale is all over the place, and now it's outbreaking up in the Northeast as well. We do get a lot of calico scale here in Kentucky. We get some tulip tree scale. And then, of course, gloomy scale is a really common pest on red maples specifically all throughout the eastern US. So, the reason we care about scale insects is because ultimately they create trees that look like this. Scales are a chronic pest that, doesn't kill a tree overnight. It's a very gradual process. But when they sit on a tree and proliferate over time, you end up with really unhappy trees that look like this. Tons of dieback, some dead wood in the canopy. And then eventually this becomes a hazard over time because you have dead branches that can fall on people. Also, obviously doesn't look good from an aesthetic perspective. If a tree is infested with soft scales, they get a lot of honeydew dripping from those trees, which people don't like for various reasons. And then, ultimately, it's also a lot of work to treat a scale infestation. And when a tree gets to this point, they're, getting close to that point of no return where you might not even be able to resuscitate them, even with a lot of tender love and care. So oftentimes what happens is when trees get to this point, they're just cut down and usually they're not replaced. Hopefully, I hope people do replace them, but oftentimes when it's here, just like a parking lot, usually it's easier for the business that owns that parking lot to just plant grass and call it a day and not worry about planting another tree. So when it comes to the different types of scale insects that we have, there are many different families. I'm going to talk about the three most common ones that we see here in the eastern United States, but just know that there are other ones out there. Starting off with armored scales, they are named because they have this thick outer covering that is separate from the body of the insect itself. So this is a zoomed in photo of some gloomy scales on a red maple branch. And so that one exposed in the center there, you can see the soft fleshy body of the insect with the outer covering having been removed, whereas surrounding it are a bunch of other gloomy scales with that covering still intact. So when we say armored scales, it basically just means that there's a separate covering that is not part of the physical body of the insect. And in contrast, the other really common group of scale insects that you're likely to encounter are what we call soft scales, and these lack that detachable cover. So these are some common examples of species that we have here in the eastern US, cottony maple scale, magnolia scale, and calico scale. Um, the name can be a little bit confusing because when they're fully formed adults and when they turn into ovisex, which we'll talk about in a minute, they do get fairly brittle and kind of crunchy. So they're not necessarily soft for their entire lifespan, but when they're nymphs and when they first become adults, they can still be a little soft. So, the name is only so much helpful, but just know that it really just refers to, or an easy way to remember is that they don't have that separate covering on the outside. When it comes to the ways that soft scales damage trees, again, they produce that characteristic dieback. Now, obviously, dieback can be caused by lots of other things in landscape trees. Your next tree fund presentation on emerald ash borer, I'm sure they'll talk about this as well. It's a really common sign of emerald ash borer infestation as well. But scales can also cause dieback and create very unhappy looking trees. A common sign of soft scale infestation is the same that you would have with something like aphids, where you get a lot of that sticky honeydew dripping from the aft end of the insects, as you can see where that arrow is pointing to on the slide right there. So basically what's happening is these guys are constantly feeding on the phloem of the tree. And so they're constantly absorbing this phloem much more than they can digest. So they're constantly just pooping out little droplets of sugar water, essentially from the tree that is dripping below it and creates a nasty, sticky surface. Where that honeydew accumulates, you end up with this sooty mold, this black surface that is aesthetically displeasing, but also it causes issues with photosynthesis for the plant because if you have a lot of this all up and down the leaves of a tree, it does block the sunlight hitting the leaf surface, so it affects the ability of the plant to absorb the amount of sunlight that it needs for survival. In contrast, when we're talking about armored scales, they encrust the branches or the leaves of trees, depending on what species you're talking about. And so they still create dieback in these plants, as you can see on the right there, but they're not going to be producing honeydew. So that is a major difference between armored and soft scales, but otherwise their biology is fairly similar. And so this is an example here These are some red maples that were actually planted fairly close together here in Lexington. On the left, we've got a red maple that's clobbered with gloomy scales. You see a lot of that dieback, very patchy growth within the canopy of the tree. And then on the right there, we've got a tree with a very few gloomy scales. The canopy looks pretty happy. The tree looks pretty healthy. And that's, typically what we would expect of a red maple when nothing is affecting it. So the last scale family, and this is not the last family that exists, just the last one we're going to talk about for this presentation. These are felt scales. They're named because they have this white powdery covering on the outside of the body of the insect. Functionally, they're very similar to soft scales in that they poop out that honeydew. They create the sticky, sooty mold below the tree where that honeydew is accumulated. Crepe myrtle bark scale is probably the most obvious one that you're going to encounter in your day-to-day life if you're in a place where crepe myrtles are grown. Another somewhat common one, but it's not necessarily a pest a lot of the time, but they can be pests occasionally, are oak felt scales, which tend to really like various different species of oaks. And then down in the bottom center there, you can see what a crepe myrtle looks like when it's got a ton of that sooty mold on the trunk of the tree. So this is the generalized life cycle for scale insects. Now, I can't talk about all species because there are lots of deviations from this generalization here that I'm depicting for you. But this is generally how scales function on trees in the Northern Hemisphere, where you've got a temperate climate. And we're going to focus here on lecanium scale because this is the species that we studied for this project. But it's also a good example for lots of other species as well. So at this time of year where I'm at, it's winter time. We're getting close to the start of spring. Right now on trees are oak lecanium scales. We're starting on the far left here of this slide. They're overwintering. They're just kind of sitting on trees. They're what we call second instar nymphs, meaning that they've molted twice to get to this point. Just hanging out on trees, not really feeding, not really doing much of anything, just waiting till it gets warmer. And then once it warms up, it becomes late spring. That's when they start waking up. They start feeding. They'll molt and then continue to molt however many times, depending on the species you're looking at, until they turn into adult females, which is what we have on the top right there. So the scales become much more noticeable as they grow. So that earlier stage when they're nymphs, they're usually fairly inconspicuous and hard to find. But once they become adult females, they get really big, swell up, take up a lot of space on branches. And then these females essentially just turn into giant ovisecs or a giant collection of eggs underneath the covering or the body of the insect. So what happens is the female just turns into a giant mass of eggs. All the babies hatch, which are what we call crawlers. And so in that bottom right image, you can see the photographer has peeled away one of these females, and you can see all the little white egg cases underneath there and all the orange little crawlers crawling away from the eggs. And so they're spreading all up and down the tree. And that female basically is not alive anymore because she's basically morphed into a giant mass of eggs. Those crawlers are the only time that this species is going to move around on plants. Well, sorry, I take that back. They do move a second time, which we'll talk about in a minute. But aside from this period, this is when they move around the most. And so they're moving around, trying to find leaves to feed on. And so that's what they do during the summer. So that's on the bottom left there. That's what those first instar nymphs, those crawlers look like when they've settled down and started to feed. So they'll sit and feed on leaves over the course of the summer. And at this point, they're super duper tiny, very hard to see. You need like a hand lens to see them accurately. With oak lecanium scale in particular, you can, if you have a good trained eye, you can notice the little orange spots on the leaves, but we're talking about the size of a pinprick, so very, very little. And then they go back to the branches at the end of the season when leaf drop occurs, and that's where they'll hang out over the course of the winter. But after this point in time, they never move again, and they'll stay in place for the rest of their life. So some of the cool things about scale insects, the vast majority of them that you're going to encounter are females. Now, this varies again by species. Lots of scale insects are able of asexual reproduction, which we call parthenogenesis. These are two extremes here on this slide. Calico scales on the left, Males of this species do exist, but at least here in Kentucky, they're all females and they all reproduce asexually. Males have not been found in this part of the state, at least to my knowledge. Whereas on the right here, we've got San Jose scale, which is a really common pest of tree crops like apples. This is a species where they do have to mate to reproduce, so they are obligately sexual reproducing species. The males look very, very different from the females. The males look like tiny little gnats, super duper tiny, very hard to get a good photo of, but that's what they look like on the bottom right there. But when you're looking at a tree that's covered with scales, the vast majority of them for most species are going to be females. So scale insects do really well on urban trees for a couple of reasons, and we're going to talk about that in a minute. That's part of why my lab researches these insects is because they're far more pestiferous on landscape trees than they are in forest settings. And in the case of oak lecanium scale, this is like perfect habitat for oak lecanium scale, as we'll talk about in a minute. So one reason that scales do really well on urban trees is this effect called the urban heat island. So basically, as you move from rural settings to a more urbanized setting where you've got a lot of pavement, lots of roads and buildings and concrete, these surfaces absorb sunlight and they re-radiate that energy as heat. And so what this means is that you can actually measure a noticeable increase in temperature as you move from more rural settings to in this image here, they're showing like a central business district. And, this isn't a tremendous increase in temperature to us. We're talking about a couple of degrees Celsius change here. But for an insect, that really can be very influential for their development rate, their fecundity, the number of eggs that they lay, and several other things, which we'll talk about in a second here. So this has been looked at. So the lab that I did my PhD in, those researchers did a lot of research on this topic. And so this is the case here with gloomy scale. This species in particular produces many more eggs in trees that are much hotter in temperature. And, we're not talking about a substantial increase in temperature here, but it is enough to result in basically a doubling of the number of eggs produced by individual females on these trees. So on all of these graphs here, we're showing egg count from individual females on the y-axis and then change in temperature on the x-axis. The reason there are multiple graphs here is that they sampled gloomy scales at multiple times in early to late spring to early summer. So basically, no matter when they sampled, they always saw this slight trend here. Another reason that scales benefit from the heat island effect is they tend to develop a lot quicker, meaning that they reach maturity faster because insects are cold-blooded, so they absorb heat from their environment, which influences their metabolism. So scales develop faster. And in the case of lecanium scale in particular, they tend to survive better. What they found in this paper was about a 20% increase in survival rate of those nymphs when they first emerge and complete development. It's not exactly clear why this is the case, but nonetheless, the data seems to suggest that they do receive some sort of survival benefit. Another reason why scales benefit from urban trees is that our urban trees are very unhappy most of the time, especially when they're growing in places like this where they've got very little soil to grow in that's not compacted. You've got those heat island effects, and then obviously not enough rainwater is going to get into the soil that these trees need for survival. So what happens is when trees are stressed, they tend to move nitrogen around in the phloem to push it towards developing growth and the roots and in the branches. And so what this means is that nitrogen is moving around in the phloem. Scales are on those trees already. They're feeding on the phloem, so they get to absorb that excess nitrogen, which they get a nice fitness boost from because nitrogen is a limiting ingredient for scale insects. Phloem is predominantly carbon and a little bit of nitrogen. So by getting that nice boost of nitrogen, they kind of get more of what they want. So essentially, stressed trees are more nutritious for scales at the end of the day. So given that stress trees are better hosts for scale insects, one really easy way that you can try to address scale infestations is to try to alleviate the stress on the tree. And so the lab I did my PhD in, they had a project where they looked at putting these water bags below trees red maples infested with gloomy scale, and they did notice an improvement in the health of the tree just by giving the tree that supplemental water. By turning off that stress signal, you're reducing the amount of nitrogen moving around and the ultimate benefit that the scale insects in those trees get. Now, we don't know how effective this is across species overall, but it does seem to work with gloomy scales. And, if a tree is water stressed, regardless if it has scales in it or not, supplemental water is usually a good idea regardless. And then it's for the same reason that I talked about with nitrogen, that you want to avoid fertilizing trees that are infested with scales, because again, you're just going to give the scales more of what they want, especially if you put down a lot of nitrogen at the root zone of that tree. Now, since we're talking about things that can be fairly obvious at least when they're late instar nymphs or adults, you can scrape scales out of trees with something like steel wool. And this is an effective control strategy if you time it right. When scale crawlers have emerged, you're kind of too late because they're so tiny, you're not going to be able to scrape them out of a tree. But when they're those late instar nymphs, really big and puffy, you can go up and down the trunk of a tree or the branches and scrape them off. This is really not feasible though if you're managing several, hundreds of trees, or if you're managing trees that are really big. But if you've got a small house plant or a young sapling-sized tree in your front yard that you want to do this with, it can be pretty effective in reducing infestation pressure. When it comes to insecticide sprays, the unfortunate fact of the matter is that they just don't work that well for a variety of reasons that we're going to talk about. They can work well, but I would say it's almost 50-50 in some instances if your application is going to work well or not. And there's several factors that influence that. If you look at this picture here, this is a person treating a tree for calico scale. They're spraying an insecticide into the canopy of the tree. And if you do your timing right, so typically you want to apply sprays when those crawlers are emerging, because that's when they're most susceptible to insecticides, you can get good control. The problem is, though, that you really need full canopy coverage, which again just becomes more and more of an issue the bigger of a tree that you're dealing with. And we also have to contend with the fact that we're talking about very, very, very tiny little insects. This image on the bottom right here is showing veins on a leaf of a tree with a bunch of little calico scales on there. There's super duper tiny insects. And as you can imagine, in a tree of this size, which isn't super big in the grand scheme of things, there's lots and lots of hidey holes where they can avoid that spray and avoid contact with the insecticide. So a lot of folks will prefer to use a systemic insecticide like our neonicotinoids for scale insect control. And the benefit here is that there's a lot of ways you can apply it, and one simple way is just to mix it in a bucket and pour it around the base of the tree. And these can work well, but the problem is that the efficacy can be really quite variable for a slew of factors that we don't fully understand the breadth of, unfortunately. But one benefit with this approach is that you don't really have to precisely time when you apply these treatments, because it gets all throughout the vascular system of the tree so that the scale insects will absorb it as they feed on the tree. And so this is some data here from another project from one of my other grad students. As part of this project, we treated trees with trunk-injected dinotefuran, one of our neonicotinoids, and we also looked at foliar sprays of insecticidal soap. These were made by licensed arborists. We didn't do these ourselves because we wanted experts to do it. And so we looked at these trees 1 year after they were treated. We also have data from before they were treated, but I'm just not showing that here for the sake of brevity. So this is 1 year after we treated the trees according to the best guidelines that we could follow and optimal timing. And what we found is that the number of nymphs on these tree branches the following year were not significantly different from the control treatments. So those are the trees where we just didn't do anything at all. So, I'm not saying these treatments don't work all the time, but they can be really quite variable due to a whole slew of factors. Neonicotinoids are also have a lot of other issues with them. The biggest problem that a lot of folks are concerned about is that they can end up in the pollen and nectar of treated trees or shrubs. And unfortunately, even if you treat these plants after they're finished blooming, depending on what product you're using, they can still show up in the pollen and the nectar at concentrations that can be differ detrimental to the fitness of bees. May not necessarily kill them, but it may affect their fitness in other ways, like their foraging behavior, the number of eggs they lay, and a slew of other things. So this is kind of an ongoing area of research that other folks are looking at, and it really depends a lot on factors like timing, the product that you use. Dinotefuran seems to have more of these effects than imidacloprid if you apply it after plants bloom. But it also depends on the plant species you're treating as well. It seems like there's a lot of factors going on here that folks are still wrapping their heads around. And then, another problem with neonicotinoids is that they are becoming more and more restricted for use, especially in places like the European Union. They've been heavily restricted. Here in the US, that's less of the case, but it really is a state by state thing. Some states, they are very restricted for uses in residential areas on landscape trees. Other states, it's not really a problem. Here in Kentucky, there's not really that many restrictions in place, but in states like New Jersey or Vermont, a lot of your northeastern states have more stringent rules on this, and other countries have their own restrictions. So that can also be a hindrance to even using these products in the first place. So, these are the difficulties when it comes to managing scale insects. And so, the good news, though, is that lots of things like to kill scale insects. And so, a lot of the work that we do in my lab is looking at ways to increase the activity of these beneficial insects, what we call natural enemies. That's just a collective term for our predators and parasitoids. We try to find ways to support the activity of these guys so that they can manage scale insects and ultimately reduce the need to treat trees with insecticides. So things like parasitoid wasps develop on the inside of scales and then emerge when they've completed development. Lady beetles, lacewing larvae, and minute pirate bugs are some examples of predators that kill other things in trees, but they also like to feed on scale insects as well. I know in these far to right pictures, they're not eating scale insects, but at least I was able to find a photo of a lady beetle chomping on a lecanium scale. The problem, though, is that ants tend to get in the way of these natural enemies. They actually like the honeydew that scale insects produce. And so, as a result of that, ants want to protect that food resource. And so, they will prevent things from killing scale insects. You've probably seen this in your garden on things like oleander aphids or aphids on, I don't know, whatever plant you might find. There's lots of great photos of this behavior online. So basically, ants want to protect that resource. They're going to kill things that are going to try and destroy that resource. And so, you can find some cool photos online, like on the bottom right here. That's an ant chomping down on a parasitoid wasp that was trying to parasitize these psyllids here. And on the top right, that's an ant attacking a ladybug while it's in the process of trying to chomp down on some aphids. What's cool though is that if we keep ants out of trees, there is research showing that that effect tends to increase the rate at which natural enemies kill scale insects. So it makes sense if you stop the guy that's blocking the other guys from killing the pest, you tend to see more of that predation and parasitism occurring. So this has worked from my predecessor here at UK, Dr. Dan Potter. He had a grad student back in the 2000s that did a project with calico scale and magnolia scale in landscape trees. And so this graph here is showing you trees where they excluded ants by using sticky bands on the trunk of the tree versus trees that they did not do these treatments with. And what they found is that generally speaking, at the end of the season, the trees where they put those sticky bands on the trunk of the tree tended to have far fewer nymphs on the leaves relative to the trees where they didn't do anything with. So that's really cool. It seems to suggest that, okay, we should try to find ways to prevent those ants from getting into the tree. The problem, though, is that if you put tanglefoot, which is oftentimes what a lot of arborists are familiar with, as this is used in places where you've got issues with like spotted lanternfly on tree trunks, and you want to stop it from crawling up tree trunks. It can also stop a whole slew of other insects from crawling up tree trunks, like ants. The problem with it is it's just a total mess to use. Anybody that's used this stuff in the past will tell you how much of a pain in the butt it is to work with. So that's one problem in of itself. It's just such a total mess that it gets all over your hands and fingers and whatnot. It's also not good for wildlife. There's, some really nasty photos online, unfortunately, of birds getting stuck to this stuff, or squirrels or whatever. So wildlife can get stuck to it, and it can obviously very much so hurt wildlife that gets stuck on it. And the other problem too is if you're doing this in a residential area, you always have to worry about, like kids getting their hands stuck on it, or people just don't like how it looks at the end of the day as well. So there's a lot of potential issues with using this product, even though it can be effective for keeping ants out of trees. This is my crude drawing here of, trying to illustrate an ant bridge, but ants are such voracious um, uh, seekers of food when they've got a source that they're trying to find, that they will basically sacrifice themselves to create a body bridge over these tanglefoot bands. In this instance here, we just have a yellow sticky card, but it's essentially the same idea. They'll just kind of get stuck on there, and next one will follow, get stuck on there, and then they'll create a bridge that they can run up and down with. And so ultimately, you have to replace these on a regular interval if you want to really strengthen that biocontrol effect in the canopy of the tree. And also, I should mention too, if your band isn't super tight on the trunk of the tree, they will try their hardest to find a little path on the underside of that sticky band that you construct. So with this project, we were looking to see if we could use insecticides applied at the base of trees as a way of keeping ants out of tree canopies and ultimately increasing scale biocontrol. The pros here are that there's no aesthetic issues. You're using a very small amount of insecticide, and you're not creating those sticky bands on the trunk of the tree. So you don't have that sticky mess to contend with. It also means that the process of applying it is much simpler at the end of the day. And, this is generally compatible with pollinators. Now, I don't have actual data to support this, but this is just my own intuition that, pollinators are probably not hanging out at the base of the trunk of the tree. They're going to be up in the canopy of the tree where the flowers are. So hopefully this would vastly limit exposure that pollinators and other beneficial insects would have to the insecticide in comparison to a total canopy spray or even a soil drench or a trunk injection of a neonicotinoid. There's some cons with this, and a lot of the cons are just the fact that there's a lot unknown with how to best optimize an approach like this. Depending on what product you use, it's unclear how long any protection you might get here would last. Do I need to go out and reapply this on a monthly basis, or could I do it at the beginning of the summer and it would be good to last for the entire summer? It's also unclear like what products would be a good use for this purpose. Should I use a pyrethroid, or should I use spinosad or fipronil, or so on and so forth. There's a lot of potential products that you could use. And another thing that we're going to talk about in just a second here is that the product that you might use might not work on the ant species that you have crawling up and down on the trunk of that tree. And the reason I bring this up is that, again, I'm pulling from that earlier project that Dr. Potter did here at UK back in the day. We have lots of ants that will feed on the honeydew produced by scale insects. So this table here is showing you some different infestations. So they've got on the top bar there, 8 different magnolia scale infestations, and then 6 different calico scale infestations. And those pluses indicate the ant species that they found at each of those infestations feeding on scale honeydew. And so ultimately we find that it's not just it's one ant species that's going up and down these tree trunks. It's actually many different species. And so it stands to reason that, well, your insecticide might not work on all of those species just due to random quirks of their physiology that gives them some level of innate resistance to those pesticides. So for this project, we did this project on oak lecanium scale and European fruit lecanium. I'm going to refer to these as oak lecanium scale going forward because both of them are really common in oak trees. They do co-occur and they're very difficult to distinguish from one another. And so generally speaking, some folks say that oak lecanium scale has a little bit more of these bump-like protuberances on the top of the body, whereas European fruit lecanium doesn't. But generally speaking, their biology is very similar. They're active at the same time of year. So most folks just kind of lump them together as one species, even though technically they are two different species. And so we did this project on swamp white oaks planted in landscape settings on UK's campus, as well as a horse farm here in Lexington called Mount Brilliant. Okay, and so for our study design here, we had 5 different treatments. We used a liquid ant bait that had spinosad in it. The brand name here is Antix. So we put that below the tree in these bait stations. So these bait stations are set up in such a way that the insecticide is in that cup part in the top, and then the ants can feed on the beads of honey or of the pesticide as it leaks out from those holes in the bottom. And so we put out 2 of those underneath trees that we treated with the ant bait. We also did some trunk sprays with spinosad. So that's Zoe here out in the field doing that on one of her trees. The product that we use for this is Entrust SC. We use spinosad because it's a little more compatible with natural enemies. It's also an organic product in comparison to our pyrethroid product, which I'll talk about in a second here. So we used a bifenthrin trunk spray as our other trunk spray product. So this was Onyx Pro that we used for this purpose. Again, the contrast here being that bifenthrin is much more broad spectrum. It has more repellency effects than spinosad does, but it's also less compatible with pollinators and natural enemies. And then we used Disodium Octaborate Tetrahydrate bait. Usually people just call it DOT, liquid ant bait. It's essentially a type of borate bait. So you might be familiar with borax or borate ant baits. And the brand name for this is Greenway Liquid Ant Killing Bait. And so for all of these, we chose 13 trees to receive one of these treatments. And then I don't have it listed here. Oh, okay, here it is. We also had 13 control trees where we just didn't do anything to them to see what happened. We applied 2 sprays below these trees, and the first spray was about 3 to 4 inches or so on the bottom, the basal 4 inches of the tree's trunk. But then we repeated that a few weeks later with a little bit of a wider band, and that's just because we really wanted to make sure that if there was an effect of these treatments, we could really see that, and we just wanted to make sure that we didn't apply too little product and didn't see any effect of what we did. So we collected a lot of scale insect data from these trees. So this is my other grad student, Ben, here on the left, demonstrating the use of a pole pruner. I'm sure everybody in this audience knows what a pole pruner is, but nonetheless, this is how we collect scale data from trees. So we collected 4 twigs per tree, 30-centimeter twigs, and we counted all these overwinter nymphs in April before we applied any treatments. And then we looked at crawlers on the leaves in June and August. So June was right before we applied those treatments, and then August was after those treatments had been put out for a few months. So we also collected data of ants. So she put sticky bands on the tree trunks to collect ants as they ran up and down the tree trunk. We counted the number of ant mounds within basically the mulch area below trees, that drip line of the tree. And then we also collected insect samples in the canopy of these trees using this thing called a funnel bead sampler. It's this crazy contraption that essentially is just a wooden lid attached to a pole with a metal funnel below it that you use to hit the leaves of trees, and then it knocks insects into a little tube of ethanol below that so that you can bring them back to the lab and see what you collected from the canopy of the tree. And so these are the data from those overwintered nymphs. The data here is on a per 30 centimeter basis. And these are the treatments here. So remember, this is before we did anything to these trees. This is just the natural infestations that were all already there to begin with. And we see, yeah, there's a bit of variation, but generally speaking, the averages are fairly similar to one another before we did anything. Here we're looking at the number of ant mounds per square meter below trees, so within that drip line underneath the canopy of the tree. And so again, we did those sprays on May 21st and June 10th, but we start to see significant differences by the week of June 16th. So this vertical bar here indicates when all of our treatments became statistically different from our control treatments in that pale orange color here. And then that difference continued up until September 16th. So 4 months later, that's when we start to see the values are reconverging once again. The number of ant mounds below the trees is once again becoming similar to the control treatment. When we look at crawler densities, so that's those first instar nymphs feeding on the leaves of the trees, we collected these data in June and August. And so we have the data here on the y-axis. This is just the number of individuals per square centimeter. And so when we look at June and August, we don't really see a statistically significant difference between the treatments. The treatments are very similar to one another in both cases. But what is interesting is that overall the densities decreased in August. And my guess is that this is just due to the effect of predation and parasitism that naturally occurs out in the wild. Just things are eating these little guys after they've settled on the trees. But at least here, it doesn't look like those treatments were doing much of anything. What is really cool, though, is that when we looked at parasitism in in August of those crawlers, we did see a significant effect of our treatments and such that our Entrust SC, so that's our spinosad spray to the base of the tree trunk, that's the only treatment that was statistically significantly different from the control. So that's what those letters mean above those bars there. Different letters indicate a significant difference. So with the Entrust treatment, we're talking about just a little less than 30% parasitism, whereas with the control treatment, we're talking a little less than 10%. Now, the other treatments were greater than the control, but they weren't statistically significant. So I can't say, with certainty that they themselves are contributing to that effect. Believe it or not, parasitoids can complete their entire life cycle within one of these tiny little crawlers on a leaf. And so that's what that arrow is pointing to there. That's a dead crawler where a parasitoid wasp has fed on the insect side of the scale and then emerged to go off and do its thing. So that's the type of stuff that we're counting in those trees. Now, this is a complicated graph, so don't get too overwhelmed. We'll walk through it bit by bit. These are the data from our sticky bands that we put around the trunks of the trees at each of the dates that we collected these samples from. I will note that our last 3 or 4 samples have not been fully processed yet, so we're kind of stopping before the end of the experiment here just because Zoe is still identifying lots of these ants. But what we see here is that starting off on June 4th, so remember we did our sprays on May 21st and June 10th, we're starting to see some differences in the means, not a significant difference, but some separation between our bifenthrin spray, that Onyx Pro, and the control treatment. And then again, we see that same effect in June 17th. When we get a little further on, this is where we get a difference between the antics. So that's that spinosad bait and the control treatment. But again, it's still not statistically significant. And then here is where we have our first significant difference between that spinosad bait and the control treatment here. And then finally, on the last time point, we are still seeing that difference, even though it's not significantly different. So we're still processing the data here, still going to see what it looks like over the course of the whole season. Now, we collected a ton of ants from these trees, many different species, over 24,000 at the current count that we're doing. So Zoe's been hard at work identifying all of these ants. These are the four most common species that we found in these trees on a square centimeter basis, or this is per 50 square centimeters. And as you can see, there's a lot of variability between treatments, which to me suggests that these treatments are not affecting ants homogeneously across the board. It really seems like certain species are more affected over others to certain treatments. So that's something that we're going to look into and do some more analyses on in the future. We also, this is not a complete project yet. We're going to look at those overwintered nymphs in a few weeks here once we get into late March to early April. That will tell us the effect of our treatments on the number of scales that survived and made it through the winter. So it'll be another time point where we can look at parasitism and the number of surviving scales so we can really see the full impact of our treatments roughly 1 year after we applied them. We also collected a lot of natural enemies from the canopies of these trees, and those identifications are still forthcoming. So it may be the case that some of our treatments were better able to support the activity of these natural enemies, and we may have seen more of them in trees treated with some of our products versus others, but we're still just in the stage of identifying right now. In 2026, we are planning to continue this project, and one thing we are interested in is combining some of our treatments. So we're still debating the exact way we're going to do this, but right now we're thinking that we're going to try combining the spinosad sprays with our DOT bait because they're two different modes of action. They probably affect different subsets of the ant community. And so we might get greater efficacy by combining them rather than doing one or either alone. And then we're going to compare that to each of these treatments by itself. And then also we'll have some control trees. So overall, I wish I had more, a more definitive answer for you all about how effective this strategy is, but such is the nature of research. We found that these spinosad trunk sprays did result in an increase in crawler parasitism. But from our current data set, it's really unclear how often an arborist would have to spray trees to get this effect, how many sprays are necessary over the course of the season, or how far apart those sprays would be. It's also unclear of like how long after you do this are you going to see an improvement in the health of the tree. Obviously, by killing more of the scales, you're going to have an improvement in the health of the tree, but it just might take a long time because, as we know, these effects are very gradual when trees respond or and improve after a really bad pest infestation. So we still got a lot to go with this project. As I mentioned, looking at what happened 1 year post-treatment, how the different natural enemy species and ant species are affected is something we're looking into, and we're thinking about combining some of these treatments to see if we get stronger results. So here at the end, I got a lot of people to thank. Of course, I had to thank the Tree Fund for funding this project. It's been greatly appreciated. And, without the support of you guys, this project would not be possible. This project was also funded by funds from the USDA Hatch Act as well. So I'm grateful for that. Zoe did all the field work for this project. So, Zoe's a very hardworking student and has done a lot of great work. And I think that's very evident from the work results that you've seen so far. We have a lot of really great technicians and undergrads in the lab, and my predecessor here at UK, Dan Potter, gave us some really great advice for this project that I was really grateful for. Several of these products, all of these products, were donated by the companies the brand names that we used. So I'm grateful for their support for donating all of these products. And then I'm also thankful for UK Grounds and Mount Brilliant Farm, the staff at these locations that gave us permission to conduct this study and helped us with, some of the day-to-day stuff in terms of finding trees and so on and so forth. If you're interested in learning more about the work that we do in my lab, this is my lab website. You can also send me an email or give me a call, and I'm happy to chat with you about any questions you may have. And I don't have a last slide here, so I'll just stop here and we can stop for questions with the time remaining. and I'll just make this
Dr.
visible.
Wilson, thank you for fantastic presentation. Zoe, great work in the field for this study. Very cool to see the results of that. We do have a number of questions, and so I'll jump in and start kind of asking away. For all of y'all listening, if you have some questions, please use that Q&A function. You can add those questions there, and with the time permitting, I'll ask away at least until the top of the hour, so about 6 minutes or so. So the first
Sure.
question I'll throw your way, Dr. Wilson, is um, have studies on nymph survival on hotter trees might somebody asked if it might be due to predator suppression.
Oh, yeah. Well, so that certainly could be the case. So some folks from Steve's lab, so this is work from Steve Frank's lab at NC State, if you're interested in learning more about what they've done. They have, to a certain extent, looked at the effect of like if hotter trees are just way less hospitable to natural enemies. And the answer is kind of, it's a little complicated. That's real world, that's the scientist's favorite answer to everything, I'm sure. But it seems like certain taxa, like in the case of one project, they looked at spiders in particular, and certain spiders do not really seem to do well in really hot trees, but others don't really seem to be affected. So it's not the case that overall natural enemies are going to be less abundant in hot trees, but certain species vary in how much they can tolerate those hotter temperatures. And so I would suspect that that probably does contribute to a certain extent to that effect of scale survival. I believe that project that I was pulling from there, they were actually doing it in like actual climate chambers. So they were isolating that effect. But out in the field, out in the real world, that certainly is the case. They also have a really interesting paper from that lab where they showed that it seems like lecanium scale develops faster than a lot of the parasitoids that would parasitize it out in hot urban trees in Raleigh, North Carolina. So basically, the scale is developing past the point at which it's susceptible to parasitoids that would normally emerge at a time of year when they would be susceptible. So you kind of get this mismatch between when the scale can be parasitized and when the parasitoids are active as a result of that heat island effect. So, it gets really complicated, but there's a lot of really interesting ecology going on there.
That's fascinating and something that's I think going to continue to rise to the surface.
Mhm.
Another question: somebody asked about the efficacy of imamectin benzoate.
Yeah, I don't know of many studies that have looked at imamectin benzoate. That's something I have not investigated myself. Whoever's got that question, if you want to send that to me, I can look into that a little bit more, but I'm not familiar with many studies that have actually tested it as a product. So my guess is that people have tested it, find that it doesn't do anything, and then they don't publish it. And so then, we just don't hear about it. But my guess is that it probably doesn't work that well. But I've been wrong before. So to be honest, I don't know off the top of my head.
Thank you. And there was two or three questions asking about maybe some comments from you related to dormant oils or neem oils for some of this for
Yeah,
control of some of it.
yeah, yep. So dormant oils or summer oils either can work well for scales. Again, depending on when you're treating the trees, the timing matters a lot. And some people have find good efficacy. Some people don't. It's another one of those issues where they can work well, just like you could do a foliar spray of something like bifenthrin and it can work well, but there's lots of instances where people do everything right and it doesn't work well. So um, I have to remember this correctly. I always get this confused in my head, but there's a paper that looked at dormant oils, or maybe they were summer oils. I think they were, anyhow, they were using they were looking at insecticidal soap and horticultural oils. And if I'm remembering correctly, the horticultural oils worked better for armored scales and the insecticidal soaps worked better for soft scales. So it seems to also matter about the actual the chemical composition of the exoskeleton of the insect matters with how well these things break apart the outer covering or how well they clog the spiracles and cause that suffocation effect. But, even if you do all that perfectly, there's also this problem of just getting to all the nooks and crannies where they might be on a tree. So again, with the dormant oil, you've got the benefit of all the leaves being off the tree, so that's a little bit easier to get all the different spots, but you can still miss plenty of locations where they might be hanging out. So yeah, great question.
especially as you're looking at larger mature trees.
Yeah, but I mean, like people treating for like San Jose scale and like fruit orchards and stuff, they do that a lot in the winter months for that pest, but they usually still have to do supplemental treatments because that's just usually not enough by its own, unfortunately.
There was another question here that somebody asked about. They've noticed that trees with girdling roots have scale problems,
Uh,
and trees with root flares do not. And they wonder if sap flow or sap flow speed might be a factor.
Yeah, that's really interesting. To be honest, I have no idea, but it certainly would be a cool hypothesis to, test one day. I mean, the other factor too is that I imagine a tree with girdled roots is probably going to be a little more stressed and more unhappy, and generally as not, I mean, I can't speak in absolutes, but usually more stressed trees are more attracted to scales as well. So I think it's like a lot of interacting factors at play, but certainly that could be part of it. So yeah, good point.
And I apologize, everyone. A lot of other great questions come in here. What I'll do is I will send these questions to Dr. Wilson or Heath will with Tree Fund. And if he has time, maybe he can look at some of those and maybe we'll post some of those responses.
Thank you.
No promises there, but we will consider doing that. But we are at the top of the hour. I really appreciate you, Dr. Wilson, and Zoe joining us for this webinar. Great piece of research. Great to see some of the work that Tree Fund is doing to support our boar culture through the work that you all are doing at the University of Kentucky. So thank you for joining us. To
Yeah,
all
thanks for
our
having me.
attendees, thank you. We had a great number of people here from a lot of different places. And as Heath noted, just a reminder, Heath, we'll be back with Rich Hauer when?
April 21st.
Awesome. April 21st. We always know that Dr. Hauer always does a good presentation as well. So with that, thank you. And just I'll leave this as a closing note. Remember to support Tree Fund. Heath put that QR code to support. These are the types of research that Tree Fund is able to do with support from arborists like you out there. So keep up the good work. Hope we have spring right around the corner and see everybody in March or in April. Have a great
Yep.
rest of your day.
Thank you.